In this video we share the main findings from our work investigating early human lung immune cell development and its role in epithelial cell fate. Previous studies of human lung development have focused on epithelial and mesenchymal cell types, but much less is known about the developing lung immune cells, even though the airways are a major site of mucosal immunity after birth. An unanswered question is whether tissue-resident immune cells play a role in shaping the tissue as it develops in utero. For our study, we used samples aged between 5 and 20 to post-conception weeks, which spans the embryonic, pseudoglandular, and canalicular stages of lung development. We isolated CD45 positive immune cells from human fetal lung tissue via digestion and FACS and used them for single-cell RNA sequencing, site-seq, and TCR and BCR analysis. We also used lung tissue for immunohistochemistry and single-molecule FISH to characterize and validate specific immune cells. And we used embryonic lung samples to generate lung organoids for studying immune cell epithelial communication. Using 10x genomics 5' single-cell RNA-seq, we obtained nearly 80,000 high-quality transcriptomic profiles, which covered 59 cell clusters, including all the known leukocyte lineages and their early progenitors. We saw significant differences in cell type proportions with fetal age. At the embryonic stage of lung development, we observed an early wave of innate immune cells, including innate lymphoid cells, natural killer cells, myeloid cells, and lineage progenitors. By the canalicular stage, we detected naive T lymphocytes expressing high levels of cytotoxicity genes, and the presence of mature B lymphocytes, including B1 cells. Our single-cell data showed that all stages of B cell development, from pre-pro B through to mature B cells, were present in fetal lungs. Combining B cell trajectory analysis with single-molecule FISH and immunohistochemistry, which showed clusters of B cells from different developmental stages in perivascular regions, we deduced that fetal lungs provide a niche for full B cell maturation. Given immune cells' presence and diversity during development, we investigated their possible effect on epithelial maturation. Using a lung organoid model, we measured the effects of several cytokines on SOX9 and SOX2 expression in organoids using real-time PCR. IL-1 beta was identified as a cytokine of interest and was shown to cause a significant decrease in SOX9, increase in SOX2, an increase in basal cell differentiation markers TP63 and KRT5 at both the transcript and protein level. In vivo, using IHC, we were able to show immune cells located throughout the lung, including adjacent to SOX9 positive epithelial tip progenitors. Our RNA scope showed immune cells near the epithelium expressing IL-1 beta. Based on our organoid experiments and this staining data, we proposed a model in vivo whereby immune cells secrete IL-1 beta, which promotes epithelial tip progenitor cell exit from a self-renewing state by downregulating SOX9 and increasing SOX2, and increases the expression of TP63 and keratin5, which promote differentiation to basal cells. Our single-cell dataset showed that the top 5 highest IL-1 beta expressing cell types were all myeloid. Looking at those myeloid cells in vivo, we found DC2 dendritic cells and macrophages throughout the lung, and also in direct contact with SOX9 positive epithelial tip progenitors. We isolated dendritic cells and macrophages from fetal lung tissue using FACS and probed cytokine rays with their culture supernatant, and found that both cell types secreted IL-1 beta. Therefore, our data suggests that fetal lung-derived immune cells have the ability to direct the developing epithelium. As far as we know, we are the first to show that immune cells are able to contribute to cell fate decisions during human organ development.